So I wasn't quite sure exactly how to pitch it, how much biology to do, how much, to describe the kind of, learning models that we use. But I sort of I assumed that there will be more people in this that would perhaps know the computing part of it. What I decided I'm going to do is describe the biological questions and domains. And it's actually both very interesting and it's very topical. And I've been sort of working on this now for 8-9 years, and I'm actually myself surprised at how well some of this, learning works. So that's why the biological context is going to be useful. Okay, so that is what we find. There's always the question: how do we know that what we learn is really, in this case, biologically relevant? So that's what we're going to talk about. So let me share my screen, and so this is, make sure you can see the screen, right?
Yes.
Okay, all right. So, so the question that we asked after, or so the path we're trying to take is to really understand the kind of functional code that is embedded in the human genome, right? So we sequenced the human genome 20 years ago, and initially it was 1. Now there are clearly over 1 million, not all publicly available, but and the question is that, there's a sequence. The sequence is, sometimes it's hyped, but still the sequence is the basis, or it's the blueprint of being a human being. And the variation between us, we know there's huge amounts of evidence that leads to the phenotypic difference. It's not the only reason why we are phenotypically different. There are obviously components of the environment as well. But the sequence, again, is turning out to be one of the routes by which the outside communicates with the inside, okay? So our interest is all the way from molecules to traits, and which is why there's this graphical abstract. We're interested in the sequence, how the sequence gets translated to some kind of active, in this case, pathway or network, how this leads to really the distribution of a phenotype across. This shows something that has a sex difference and really then finds expression in not only that individual, but how it's transmitted within families. And we sort of try to cover that whole ground. So just to give you a sense that much of genetics really for more than 100 years has really focused on trying to understand the functions of individual genes that specify proteins which when mutant lead to a disease or some trait. It doesn't have to be a disease. So, but very early part of the 20th century, so there's Ronald Fisher, many of you would know as clearly one of the fathers of modern statistics in the 20th century, and Hermann Muller, who was really a very famous Nobel Prize winning geneticist who worked on flies and who in fact found the mutagenic basis of X-rays and how it changed biology. That there was work by both of them. I'm not going to read all of this because if you're going to record it, you can read this in detail. But Fisher was the first who suggested how a big genome that had many genes, how all of the genes in fact could have effects that are additive across all of these genes to produce, for example, the normal looking distribution of a phenotype. So this is, say, the population distribution of height, weight, cholesterol, blood glucose, anything else. If it's a qualitative trait, it's viewed as those that have one kind of phenotype versus the other. It's the threshold. But this distribution is because of the effects of many genes, of course, the environment as well. And the fact that it was the case was considered originally to be really, this was not taken very seriously for many years because we didn't quite have the tools. But Muller really did this paper where he showed something that had an unusual inheritance called truncate wing, in fact, could be divided, okay, into those three or more sort of factors. So this has sort of been an added, meaning, theme in my lab for a very long period of time, well, I'd say now close to 30 years, that is starting with this disorder, which I thought was the template at a time when in genetics we were only looking at single gene traits or single gene disorders like cystic fibrosis, muscular dystrophy. And I'd always been interested in how this multiplicity, how do multiple effects get integrated, okay? So that's really the question, because that's really the problem of the genome. The problem of the genome is to not do things one at a time in some, uncoordinated way, but to do many, many things in a very coordinated way. So I don't want to talk about the disease, but the disease is actually, it's a neurodevelopmental disorder of the gut, where your gut doesn't get, a nerve supply doesn't get innervated, and that leads to a kind of functional block in the intestine, and in fact has to be surgically repaired. Otherwise, it's a lethal disorder. So before surgical repair in the 1950s, this was uniformly lethal. The individual didn't survive. Surgery actually brought forward the fact these individuals could now survive and reproduce. And so that gave us much of the genetic material to be able to study. While this was going on, and by the way, we were discovering genes. In fact, my group was one of the leading ones where we discovered many genes, but we had an observation that despite really beginning to understand the biology, where we did expect mutations in a gene, we never found them, even though we knew from other evidence that the genetic defect mapped to that locus, not the only one. So after the sequencing of the human genome, there were a number of us. This was sort of an early paper. So this is shortly after the sequencing of the human genome, and doing these kinds of studies were difficult. We knew that genes were conserved, but this was one of the first evidence that there was widespread sequence conservation outside genes. Okay, so this shows a portion of a locus. The y-axis in each individual panel is in fact some kind of a running window in which the sequence is conserved. So when you look at this, these are all comparisons in this region. This is, by the way, so this is the cystic fibrosis region. So it shows, for example, this one, this particular segment, that if you look at humans and chimpanzee comparisons, almost everything is conserved. Okay, the sequence is almost identical. When you look at baboons, human baboons, you find there are little bits of changes in the conservation. It's more blocky. As you go to other mammals, it becomes, conservation is now restricted to small segments, and you can go all the way to zebrafish, right? So this is what shows the unity of both development and evolution. You can go to zebrafish, and here are this small segment that has survived over 500 million years of evolution on each side of the branch. So to me, this is sort of remarkable. And anyway, we discovered this, and this was, in fact, the beginning of our work in thinking that we were already doing directed work because we knew of this kind of stuff, meaning early results, that this must be one reason why, when we know that a gene is involved, that we have to search beyond, the functional known coding portions of the genome in order to find mutations, because there are regions outside the gene that do control the gene that are regulatory and have mutations. That's clearly the case. Again, I don't want to, belabor this point, but we did find a region, a genetic element, a piece of sequence that was highly conserved. In this case, it shows mouse-human conservation. All the red parts are the coding sequences, but we found the sequence, the blue are the coding sequences. Here's exon 1. The red are regions that sit outside coding sequences that were highly conserved. And we found a very suspicious looking genetic change in this one. And this turns out to be a regulatory region, because you can take it, you can make transgenic mice, and what you find in the gut disorder that I told you about, that this element, which is from the human, can drive the same regulatory activity in the mouse. And by the way, our common ancestor to the mouse is 80 million years ago. So this signal has survived. And We later found it was a regulatory sequence. It binds a transcription factor. That's how these regulators work. And what they do is they control gene expression of this. This mutation is very, very common in the world. And since then, we've done a lot of studies. I should have corrected this. This is a paper that just appeared in Genome Research. In fact, it's in this month's issue. And now the technology is so much better, we can do this. So here's a region of the human genome on chromosome 10. Here is the gene that I was telling you about. Here is that suspicious variant. And one of the things we've now defined is a whole series of these cis-regulatory elements called enhancers. We can discover many of them. I'm not going to go into the details of the kinds of data that gives other kinds of such regulatory elements, but here are enhancers. And what we identified in this red, some of this red includes two adjacent such elements. So there are these discrete elements that, in fact, show that they control expression of these genes, okay? So we can now do this in much better ways. We can clone out these species, we can express this in cells, and we know from our disease studies that when we find a non-coding sequence, which we statistically, that is epidemiologically, can show that they lead to risk. So the yellow here is the risk variant, the blue is the non-risk variant. Then there are clearly cases, just like I showed you in the mouse. There are many such elements, 10 described here, that we can do functional assays and decide and find that they do in fact regulate expression of that gene called RET. So what surprised me, and we can tell you, we can discuss later why it surprised me. What surprised me is just how ubiquitous these were. And this is just further evidence, this is from this new paper, that if you delete them, you can show that what they do is they lead to reduced expression of the gene, okay? So you can think of the gene, but think of outside the gene or even within the gene, there are these many, many sequences that bind transcription factors and regulate how much of the gene product is made. Okay, so they're like switches. And the switches sometimes act in only one tissue, but other times the switches act in many tissues. Even when you consider a single gene, I was just showing you this, that gene does not exist on its own. In fact, in many ways, it interacts with many, many other genes. And the fact that these systems exist have been known for a while. Eric Davidson is really the pioneer who started thinking in this way. And this just shows the system in ours. I'm not going to go into the detail. I want to really get to the statistical problem. So what I want you to think about is here's a gene, here's a coding sequence. Outside the coding sequence, sometimes within the gene, there are these elements. The cis-regulatory elements or enhancers, they bind specific transcription factors. These are the ones that bind. The transcription factors affect two genes that are co-regulated, and that's how a network is formed, okay? These are not the only components of the network, but we can reduce, at least as a very first approximation, the effect of every gene or the functions of every gene into this triad or this trio. So here's the target gene whose expression is determined by these sequences called enhancers. I'm going to refer to them as cis-regulatory elements or CREs, and they bind very specific proteins called transcription factors. So in order to understand this gene, you need these transcription factors that are expressed elsewhere in the genome, these cis elements to which they bind, and they come together and they control transcription of this gene. I want you to understand one thing. This gene often doesn't have to be. This gene can be tissue specific, that is only expressed in the gut, or it could be ubiquitous, it's expressed in every tissue. These enhancers can also be both, but they provide far more tissue specificity. And these transcription factors can also be both, but it's the combination, but these can also have tissue specificity. So basically the specificity of where a gene is expressed is provided by some kind of a combinatorial code, or where the transcription factors are expressed, and where these enhancers are open. They have to be open in order to bind these factors. Okay, and they lead to predictions of different genetic effects and different effects on the phenotype that is on a disease if you're studying. So this success, and it's been happening over some time, led me to think that when we come with really complicated systems, and my group has been studying blood pressure genetics for a long time, we sort of thought that blood pressure would be a good paradigm to understand. By the way, I'm not a very big fan of complexity, but I thought blood pressure would be a system where we could dissect the complexity to understand what is it that is complex and how is this complexity really coded. So I'm going to talk about building. So this, to me, has to be sequence-based, not just a descriptive saying, here's an enhancer, here's a factor, but we want to know the sequence to which the transcription factor bind, so that if the sequence changes, say between you and me, we know it will lead to changes in the gene. And that's how we are going to predict. And so this will also going to give us insight into the cardiac system, but blood pressure is important because the cardiac system is not the only tissue or system that controls blood pressure. Blood pressure is controlled by many, is controlled by the kidney, by the adrenal system, by the brain, and clearly by the vasculature and perhaps other tissues as well. So much of this thinking came about, as I was saying at the beginning, it's no use having a good thought or even an interesting idea. You need people to be able to do it. And Dongwon Lee, I interacted with him on his dissertation committee. He was a student in biomedical engineering in Hopkins. He actually had a lot of computing experience in Taiwan. He worked in industry, but he came to do work with Mike Beer. And Mike is known in this area and was one of my recruits in Hopkins. And I just convinced Dongwon that rather than doing the usual kind of thing that almost everybody else does, I said, you should really work on human phenotypes and disease, because even though there's not a lot of data, there will be. So he was a postdoc with me, and then he came to NYU with me in 2019, got a job in Harvard and in Boston Children's. So he's really been a very major architect of doing this. So here's the method. This is the method was originally published by work that Dongwon did with Rachel Karchin and Mike Beer in Hopkins, and then the other papers, and my contribution really comes on the trait and the disease part. So for those of you who do AI, this should be really much more obvious. So the idea is, here's a depiction of the human genome. These peaks represent regions that are open, okay? And so we have specific kind of technologies for identifying them. The height of this is sort of a rough measure of how open a site is. So here's the genome sequence. We know it at the sequence level. That's not what I'm representing here. When a sequence is open, or when the chromatin is open, we can detect it by some assay, and there are various kinds of assays. There are direct assays of transcription factor binding called ChIP-seq. There are DNAs where you use this enzyme, and every time the chromatin is open, it chops up the DNA. That's how we recognize them. There are newer, more popular ways called ATAC-seq and such. So you can define regions that are positive, okay, or from some external data set from which we are learning. But of course, there are also regions that are negative, okay? So they're positive and negative controls, if you think, and we're going to use them to learn what the sequence vocabulary is, okay? So far, there's nothing new. The newness was that, transcription factors classically have been thought to bind to a specific sequence, right? The A's and G's and C's and T's. But over time, it's been clear that their binding is far more complex, meaning all of these have been classically represented by what's called a position weight matrix of essentially consecutive nucleotides. But it was quite clear consecutiveness is not the only criteria. It depends on how this protein binds to DNA. So there may be gaps, that is, you require sequence specificity, a very specific kind of gap, other kinds of sequence specificities. And when the sequence specificity is quite long, Dongwon's really innovation was to say, well, I'm not going to model the entire sequence because the entire sequence is going to be quite different from one transcription factor to a second and a third. But what I'm going to do is build a vocabulary of some k-mer. In this case, it shows 10 mers. And what you can then do is use its positive and negative regions to find, in this case, 10 mers that are enriched. And then one can use support vector machines, for example, to find the degree of enrichment and assign weights. And this was essentially turned out to be really very, very successful. Successful in the sense you can learn the sequence vocabulary that a particular protein uses. More so, you can do this at a cell type or a tissue level so that you know the sequence vocabularies of all of the transcription factors that bind in a particular tissue. And we can find regulatory effects that are positive as well as regulatory effects that are negative, that is activating and repressing. So this, you could do, the usual kind of classification accuracy by using area under the curve or other related, ROC, methods. And one way of doing it is to learn from some chromosomes and apply it to the others. Okay, so the human genome has, 22 autosomes and a pair of sex chromosomes. So from the same data, you can learn, you can do various other sort of standard statistical procedures. And when you do that, what it allows you to do is something, so this was sort of my sort of entry and contribution is to say, well, if you can make a model that is sequence dependent, of course, we can also model what happens when you change the sequence, right? So here's, for example, a sequence change at a particular site. The normal nucleotide or the common nucleotide is C. So if there's a change to A, I can sort of do this delta SVM modeling again and see what the difference in weight is, right? And sum it up over all possible 10 mers that occupy that space. And I can do this for the whole genome and for all such sites. I don't have to tell you this, it's sort of trivial. Not only can you do this, you can do this once for every single site in the genome for all possible changes, right? And that can even give you a sense of what is the space in which such regulatory changes can have an effect. And this just shows one of many such assays. This just shows one of the ones, where we can use both. This is delta SVM. So SVM, you understand, delta SVM is the difference in scores. So here's delta SVM in a particular cardiac model. Here's some assay by sequencing we did, quite independent technologies, and it just shows that at least, in this case, the middle is, not shown, but in fact, there's very high concordance. That is, if you take high weights, they correspond to one state, and if you take low weights, it corresponds to the complementary repressive state. So here was one of our, this came out in a study we published in 2018, right after I arrived here, that, so we said, well, then what does the genome in the heart look like? And one of the things, and I'll come to again and again, Nargis, you got to tell me how long I can speak.
We have until noon.
Okay, so I'll probably leave some 5-10 minutes for questions. Let me see. Okay. So, there was publicly available data, but we also collected data. I'm a big fan of saying, publicly available data are good, but I like more experimental control, because in any kind of learning, the more consistent you are in your experimental learning model, the better your prediction is going to be, okay? And I no longer assume that everybody who produces and publishes data produces and publishes data of high quality. It's of various quality. And that was one of our early recognition that we worked on. So anyway, so the paper is published. This is the 2018 Genome Research paper that often this is what happens. We always talk of metadata. People will say the heart. Well, the heart is not just one thing. And so often heart regions are not specified, so they're not helpful. But anyways, we collected data. And the reason why we collected data on multiple individuals for the same kind of tissue or tissue regions is these are cross-sectional maps, right? Chromatin is open or closed. We do this assay at one given point in time. And there are many reasons, as experimental error, experimental variation goes. The empirical maps are not comprehensive. We don't detect all sites that we could easily show because new experiments find new very strong sites. There's variation signal intensity at any given point in time. The sequencing depth in reality really differences. There are protocol differences. And ultimately, it turns out to be quite important. There are stochastic changes. We know transcription in this binding it's not like it binds and then there's transcription. Yes, that's the case, but it often happens in bursts, okay? That is periods of inactivity, and then somehow all of these factors come together. We have a crude idea how that happens, and transcription happens. So here's an accounting. I'm not going to read all of them, but just to point out that we observed 160,000 such sites. Okay, so remember that there are 20,000 odd genes. About half of them are expressed in any given tissue. So 10,000 cardiac genes, maybe 12,000. And there are 160,000 such observed CREs. Okay, so basically, if you divide this, there's something like 10 to 20 such elements per gene. By the way, sometimes the elements are discovered next to one another, and we still don't know whether they aren't part of some super element, okay? So I don't mean the 16 elements are all completely independent from one another. One of the advantages of a model is we observe what we observe, but we can now go in and predict other sites that should be there, right? But we did not identify, simply because of the model. And we found that the reason why they were predicted, we often don't observe, is they tend to have weaker signals. That's because they're more cell type specific. So it's very difficult sometimes for rarer cell types to be detected in an overall tissue prep. And they often, many of them, map to repeat sequences, which are very difficult to map. We found that they overlap conserved elements. In fact, the observed ones are more conserved, which is probably why we discovered them. We also find that these cis elements, the sequence of them could be used to identify transcription factors. And we found about 35 transcription factor families, and they included, I think you might say this wasn't a positive control, but this sort of had to be the case that key cardiac transcription factors that have been found in the past by doing much more directed experiments, this included all of them. Okay. So here's just to give you a sense, for those of you who don't look at transcription factors all the time, there are many classes of transcription factors. These are not all of them. We found about 78 that were enriched. And this sort of tells you the sequence logo of them, okay? So for example, here's one. Here's even another one, let me point to SOX. There are 5 SOX transcription factors. They all show enrichments, but to different degrees. They obviously have some similarity in the sequence logo, as you can see, but they're not identical. And the point is, all of the binding sites, of which there are many, many in the genome for SOX17, show a preference for this site with an A here, which is not the case for SOX6. Okay, so the sequence logo can be identified. In fact, we can find things empirically that, for example, in cell line experiments, we couldn't. So this is good, right? So we were very excited that we could do this at the tissue level across the genome using everybody's data, our data. But we required a little bit more proof of the pudding. We said, well, it's good to do it genome-wide, but the question is, how good is the prediction for any one given gene? Okay, meaning we just don't want an average good prediction. We also wanted good prediction for genes that are expressed in the heart. And this is a classic gene. It's a sodium channel called SCN5A. Many people here, for example, in cardiology, including my lab, works on SCN5A. This is very similar to the kind of enhancer assay. It's a reporter assay that in every case shows you the reference allele and the other alternate allele, reference alternate. And these two that are different suggest that the two alleles that sit in that enhancer have differential effects. I'm not going to go through the details except to show in this plot. What we are doing here is saying that if we take all the enhancers, take all the variants that we observe, can we find the critical variants that really change the expression of the gene? So this is taken from a much more recent paper in PNAS in 2019, where we looked at cardiac gene expression. Of course, this is from autopsy material. I think this is based on, in this case, for 94 individuals. So you can measure gene expression in the heart, whatever, however good they are from the point of view of autopsy material. And what you can do is say that what we predict is pretty much what we expect. And it also points out the specific variants that in fact have all of the properties that sort of drive or change the expression of this gene. So I think given this, it let us be a little bit more bold. So what I told you, if I can paraphrase, is that we now know the human genome sequence. We know segments, very discrete segments of that genome that in fact are open, represent open chromatin in that tissue that bind transcription factors whose cognate transcription factor that binds, we know its identity, and we know that a number of them near a gene can affect the transcript or gene expression of that gene. So to me, this is really getting to sequence level code. And we also know that if we change the sequence, we're going to change the gene expression. So this is prediction. You could say sequence-based prediction, de novo prediction of, it's de novo after learning of gene expression. So now we wanted to do this in a broader way and to look at regulatory variants, I should say, for a number of tissues relevant to blood pressure. And I'm going to go very, very quickly. What we've done are these 4 tissues, the adrenal, again, the heart, in this case, the left ventricle, and looking at some arterial samples and kidney. We've now updated it with others. Here are data sets that are published in this large ENCODE project that came from other sources. There's ATAC-seq. In this case, only the kidney data is from us. The other are data that we use. This was done a couple of years ago. And one of the things I want to point out to you, you'll see that there are many such data sets, or this is many compared to getting data directly from human tissues. But we found that many of them were really not appropriate for learning. And the question is, why did we say that? And what do we mean by it? So I pointed out in some early data, which was a cartoon of actual data, that if you segment and you look at any particular region of the genome, some peaks are weak, some peaks are very strong, and some are tall, some are not so tall. So there's different intensities of these cis elements. And this represents real biological activity, but also represents all of those other factors that you said, experimental variation, stochasticity, and such. So we would expect that the stronger the peak, it has more biological activity and probably more signal strength as to what the underlying sequence is. So one of the things we could do is we could take all the peaks and sort them, right? From the best peak, highest peak to the lowest peak. And if we take them and we take bins, right? Rather than looking at each one individually, because these are only very small hundreds of nucleotides, we could bin them. And then what we could do is within, and again, create some control, negative control data. And what we could do is not only run our support vector machine sort of learning for each of these data sets. But in a way, we could sort of cross-validate in the sense that what we learn here should have been learned in a previous stronger signal strength, data set. So this is work, again, this and another paper, we are currently working on with Dongwon. So it gives you some idea. So let me just focus only on one of them, which is the adrenal gland. So this is the area under the curve. And this is the peak subsets ranked in groups of, obviously it's ranked even in a smaller group, but showing, the top 5,000, 10,000, 15,000 and so on. Okay, this is not 5, this is 5,000 rather. And as you can see, the fall off of information is quite different. Okay, sometimes it's different even between multiple replicates of the same kind of experiment. And this is an empirical observation. In some case, by the way, I should point out, this is our data. The kidney data was very, very good because by that time we'd learned, okay, that how and what we had to do in order to have high quality data. And if you look at other quality data, they have much, this is published data, is of much, much varying quality. And there's no doubt, for example, in this tibial artery, these two data sets provide far more specificity than the bottom one. And I don't expect, and we don't expect, our learning performance to be the same for these two data sets and these two data sets. Okay. So Dongwon and his postdoc, has really worked very hard on, this is not, doing QC is not the glamorous part, but it's actually a very, very important part. And I would say this is actually going to be an important paper. But here's an application where one can use AI methods to essentially find out which portions of the data are reliable and which portions not. So I'm now giving you the annotation. So I'm not going to talk much more about what we did in machine learning, and here are some more annotations of it. So here are the 4 tissues. Here's the combined tissue. Here's the number of cis elements. Remember, I told you 160,000 for the heart in a previous, by these definitions. We've changed the definition somewhat. And you can ask me why. And they vary anywhere from, say, 110,000 to 140,000. The average length is about the same, and they cover more or less the same amount of the genome individually. But when you now take all these learned elements and squish them together, what you find is, obviously, this is not additive. That is, some of the elements are in a single tissue, whereas some of the elements are in multiple tissues. So here's what the data are. So this is open chromatin regions from these 4 tissues. All of you know how these Venn diagrams are represented. And the red circles are the ones that I want you to focus on because they are the larger numbers. Every other number is sort of well under 5%. Actually, they're under 5%. And you will note that these 4 on the top, the heart, left ventricle, kidney, artery, and adrenal, represent these cis elements that are open in only one tissue. And here's 16.5% are those that are open in all 4 of those tissues. So these are the 4 tissues of relevance to blood pressure. We could add a few more. But this was, by the way, there's all these other little, sputsy stuff going on, and we don't know how much of this is real, how much of this is error. But to me and to Dongwon, really, these are remarkable observations that it looks like each of these cis elements have a crudely, if I can describe, has a bipartite behavior. They're either specific to a tissue or they're open everywhere, okay? And if that's the case, learning that code actually is going to turn out to be very, very important. This, by the way, is true even in many of the other newer data sets. So this just says, by the way, so this is about open chromatin in tissue, but a gene could be, again, expressed in one tissue only, and a gene could be expressed everywhere. And now comes the interesting part of figuring out, which we are now trying to do, that if a gene is expressed only in the adrenal, you would expect its cis elements to be open only in the adrenal, right? That's the logical construct. Whereas if a gene is expressed everywhere, if it's ubiquitous, you would expect its cis elements or enhancers to have one of two modalities. Either the gene has multiple elements, each open in a different tissue, or the gene has some elements that's open everywhere. And the reason why this is important is if we are going to talk about sequence variants in these elements, and we're going to learn this code, then sometimes a gene could be expressed everywhere, but its effect on blood pressure will occur only in one tissue. Okay. So now what we can do is we can use this kind of genetic variation data that exists, as well as the total effect of these genetic variants on blood pressure. I'm showing you, in fact, in this table, only the effects on systolic blood pressure. And we are looking at only the kind of inter-individual variations. It's called heritability, but heritability here is the proportion of the additive variance that explains how much my blood pressure differs from yours. Okay, it's that variance. I'm sorry. And what you will find here is the proportion of that variance that's expressed by adrenal, heart, artery, and kidney. They all have substantial effects. Classically, in blood pressure regulation, the kidney has been considered to be the king, but it turns out there are other tissues that make significantly more contribution to this variation. And we can explain about 2/3 of the genetic variants. There are many, many, genetic variants that contribute to it. And now this just says enrichment, and all of these are highly significant effects. So this just says there's actually no doubt that the blood pressure effects come from these various tissues. They come in various ways. And we are now trying to dissect out how many of them are tissue specific and how many of them are far more ubiquitous. This is the same diagram I showed you 2 slides ago, with one fact that Dongwon really noticed, which is that a significant portion of the variation actually occurs through, through this arterial system. So all of the ones that are in purple, and of course this is ubiquitous, are effects that are in artery at least. It could be in other places because we don't know what the CREs are. And the green, for example, are in the non-artery tissue. And there's actually no doubt that beyond the unique parts, the arterial component is actually significant. And the reason why this is important is by the way, this is the same thing, except now what we've done is not taken all genetic variants, but taken genetic variants that are likely to impact on enhancer activity. And you will find all of these other elements go down, but these individual effects, actually even the ubiquitous effects go down, but the other effects go up, which is why I think we tend to now believe that genetic effects, at least for blood pressure, as we are learning, occur through individual tissues. And that to me is a very, very different kind of thinking than we've had. So I've only showed you data on essentially one kind, which is open chromatin data, but there are many other kinds of regulatory data. We can include histones that are somewhat complementary, but somewhat unique. There isn't a single code. The human genome, uses a very combinatorial code. The DNA methylation differences that we could do, James Cock here studies, but many others too. And so it would be great for us to learn how each of these biochemical sort of decorations on the human genome really affect gene expression and how this changes when the DNA sequence changes. And I really believe we can develop a quantitative epigenetic regulatory code. So I was going to say a couple more points, but I think in the interest of time, let me just stop there. I guess let me just end by saying, this is sort of interesting because the code itself is not enough. We know that when we do have disease, there are many, many changes that happen. As I showed you, the enhancers really do contribute, and sequence variants change at least their effect. The changes in local chromatin, by studying methylation, others we could study. The gene expression variation changes, which, by the way, we often, as I told you, our data came from autopsy material. But if we can learn the code, then I can predict, it's not me, then the science can predict the expected gene expression in any given individual, given their sequence, in a tissue without my observing it. Okay, that is, we are using reference data to learn to predict gene expression in a given tissue. We can do the same thing if there were observations of protein expression. That changes gene regulatory network and the interactions that changes cellular function and variation. So there are various ways of doing it with connections to many other colleagues here, but I'm going to stop here with really much of the work that I described today is really work of Dongwon Lee, and I don't have Or's name, but Or has contributed to some of the data together with Prabhu Mathialagan, and there are many others really in my lab that have contributed to these ideas over time. And anyways, let me just, I think that's it, and take some questions if you have any.